Welcome back from lunch, everyone, and we're going to start our second panel on AMR and conflict. So an old group of friends, or a group of old friends, so it's nice to be back with the three of you. Maybe I'll just say 3 sentences, which is that there's growing evidence now, and I think it's really multiplied since we started working on this 10 years ago, that contemporary conflicts are driving antimicrobial resistance. There's sort of 3 pathways that have been identified, and we'll hear more about them. The first one is kind of obviously wounds, and with inadequate debridement. Not inadequate necessarily because of inadequate surgical skills, but because of rationed time to operating theaters. For debridement having to be done every day, or even 2 days. So you can imagine how in a situation like Beirut now, people's wounds are not being debrided adequately. Inappropriate use of antibiotics. So there's kind of a clinical pathway. There's a environmental pathway where damaged infrastructures leak sewage, antibiotics, etc., into the environment. And we'll hear we'll hear more about this, I think, around environmental contamination by heavy metals from Antoine Abou Fayad, who is associate professor in the Department of Microbiology at the American University of Beirut. I imagine circumstances are difficult, to say the least, in Beirut today and these days. So we really appreciate you making it. And over to you.
Right. Can you all see my screen?
Yes.
Okay. Thank you, FINPHARM, and thanks to Anthony for the invite and the opportunity to present to you some of the work that we've been doing on the topic of AMR and conflict, and how is AMR driven in conflict. I'm gonna spare you the introduction, and I'm not gonna be preaching to the choir about what is antimicrobial resistance. I am pretty sure you guys know what is AMR, but as FINPHARM said, this has been a lot of work has been done by a group of friends that we've been working together for quite a while now, and we've been tackling this circle of bullets, shells, destruction, and superbugs. Why is it that every time there is war, we observe superbugs? We actually observe the emergence of resistant bacteria, and what can be the drivers. Now, to make things a bit more challenging for me in this presentation, I actually started with the conclusion, and I'm going to work with you one step at a time around this presentation. So if we look at what are the main drivers, or some of the main drivers of AMR in conflict, you would see there is lack of surveillance pre-conflict, during conflict, and post-conflict. There is lack of workforce, which leads to hampered detection and misidentification of microorganisms. There is a lot of substandard antibiotics and heavy metal contamination, as FINPHARM mentioned, which we will be focusing a lot more. However, we're going to tackle bits and pieces around the remaining drivers that we've discussed. So starting with lack of surveillance and the scarce sequence data that's available in the region. And by the way, I forgot to mention that my main focus in the work is on the Eastern Mediterranean region. Hence, you would hear me speaking about countries in EMRO or Eastern Mediterranean region more than speaking about global and Ukraine. And I don't have any connections in Ukraine, so I focus on the area where I have connections in. So We know there is lack of surveillance for AMR initially in the Eastern Mediterranean region. And if you would look at pre-conflict in Lebanon, there is, we're one of the very few labs that do sequencing frequently to look at antimicrobial resistance. Egypt, it's quite controlled by the army. Not a lot of the data comes out. Saudi Arabia is doing quite a lot on sequencing. However, there is no conflict in Saudi Arabia. Sudan had a good bit of sequencing done for a very short while, but then it stopped. I mean, if you also look at the number of sequences that, let me just use my pointer so you can see what I'm pointing at. The number of sequences that you can see here. This is back in 2020 when we did this study. We looked at how many sequences were published. Now the numbers have increased, obviously, especially for quite a few countries, but you can see some countries barely have any. Like, look at Palestine, there is 31 sequences. Syria has nothing. Bahrain has a bit. There are countries that are not on the list, like Somalia, for instance, which is also suffering from conflict. And this actually poses a problem, because if we don't really understand what's the actual burden of AMR pre-conflict, we cannot really anticipate how bad it will be post-conflict. Another driver is the misidentification of microorganisms, and this is a good bit of work that we did with colleagues from Syria, where we received 2,000 samples from them to actually work on this project together from war wounded patients. And among the 2,000 pathogens, 60% were misidentified based on what they claimed they have isolated from the patient and what we have observed when we perform sequences. The 60% out of 2,000 is quite a high number, actually. I mean, even 10% out of 2,000 would be quite high. So this is something that it happens in conflict due to lack of workforce, due to the inability to perform standard tests to identify pathogens using the standard techniques that are available in clinical labs. And this is something that hampers the work quite a lot. One of the things that we have observed, and this is a piece of work that is published now, you can find the paper, it's in antimicrobial agents and chemotherapy. And this is work that we did when we were trying to look at a bunch of Acinetobacter baumannii samples from hospitals in Libya. So usually Acinetobacter baumannii, or what is also known as Iraqibacter, when you have an outbreak, if you look at the bottom side of the figure here, this is from an outbreak in hospitals in Germany, right? You would notice that each hospital would have a cluster by itself, right? Usually the outbreak would be more of a uniform outbreak, where within a hospital you would find one sequence type or one international clone. Hence, we would call it an outbreak. But when we looked at Libya, for example, we've observed polyclonal outbreaks and various distribution of all clones, looking at 3 different hospitals. And these 3 different hospitals are in 3 different locations. And these samples come from war-wounded patients from the war in Libya. This is one factor that also drives antimicrobial resistance further and actually causes the spread of various clones of highly resistant pathogens. We also observe quite a lot of the frequent pan-drug resistant bugs, which I was referring to earlier. This is one example from Klebsiella pneumoniae isolate sequence type 383 that we've isolated from a soldier in Lebanon who was wounded during conflict. And, we have actually found this isolate to have 36 AMR determinants, which is very high, pan-drug resistant, where, I mean, there was no possible treatment for that soldier aside attempting to use combination therapy where certain methods would work. In addition to the misuse and abuse of antibiotics in the Eastern Mediterranean region, and especially in the conflict areas, there is this notion of forced to use antibiotics. It's when antibiotics expiry date are dumped in conflict site to be used empirically within a few months. And this is a piece of information that we learned from our colleagues Hassan Abu Sitta and Richard Sullivan as well, when they were on their adventures, performing surgeries dire and conflictuous situations. But to add to all of this, COVID came along, and during COVID, there was a peak in the irrational use of antibiotics, especially when it comes to azithromycin and other antibiotics that were thought to treat a viral infection. Another driver of AMR in conflict is substandard antibiotics, and this is a piece of work trying to focus on for the last while. We're nearly done with the quite interesting for us to show you that when you collect antibiotics that are produced in countries that are having conflict, or that are produced in countries that export to countries that have conflict, but there is no proper regulations, you can see quite a difference in the bioactivity of certain antibiotics. And we notice usually quite a difference in the activity, twofold or fourfold. So whenever you say you have a 500 milligram pill of acipine, which is a cefepime antibiotic, quite a lot of times these antibiotics would be 200 or 250 milligrams at best. This is what we noticed. We've tested them against a bunch of pathogens, comparing them to antibiotics produced in Lebanon, in Syria, in Jordan, in Turkey, in Egypt, and now we've actually managed to get a few from Iran and Iraq and India because of the aid that's coming into Lebanon. And we've noticed the substandard quality of the antibiotics, or not even the quality, but the dosing, actually, the underdosing that actually leads and drives resistance even more when it comes to that point. And the last part that I'm going to expand on a bit more is heavy metal contamination post-conflict. How does it drive antimicrobial resistance? And this is a piece of work that with Nick and with Aula, who will be presenting right after me around this topic. And what we did, we looked at the heavy metal contamination in Lebanon post the 2006 war, and we've looked at Iraq and the publications that came out regarding the heavy metal contamination post the Iraq invasion war. And what we did, we tried to understand which heavy metals increased because of conflict, and we will tell you why in a minute. So technically, what we have is heavy metals, they work in a way where they were considered to be antibiotics pre-antibiotic era. Before the discovery of antibiotics, heavy metals were used to treat infections because they have the capacity to kill bacteria, to kill not only bacteria, but they can kill parasites as well, and work in a similar way to antibiotics. And when we start looking at mechanisms where resistance to antibiotics and resistance to heavy metals can cross path, we can look at cross resistance when you would have one gene which could encode to resistance to both antibiotics and heavy metal. And quite common is an efflux pump, which is quite good at pumping out of the bacterial cell whatever harms it, like heavy metals or antibiotics. There is the concept of co-resistance, where we would have the antibiotic and the heavy metal gene both on the same plasmid. And there is something called co-regulation, where one regulator would regulate two genes, one for the antibiotic resistance and one for the heavy metal. These are the ones that we know from the literature. These are the ones that have been a little bit studied, but not deeply enough, and not in the context of conflict, obviously. So what we did was we actually took the heavy metals that we analyzed and saw have risen from conflict, and we started also with bullets. So we've spoken to the Lebanese army. We acquired some bullets that have been fired and some shells as well, shrapnels, and we started growing bacteria Over time, in the presence of these heavy metals, looking at lead, at barium, at arsenic, and zinc, at chromium, at cadmium, at copper, and mercury. And we tried to actually make the bacteria resistant to these metals by doing several passages. So you would incubate the bacteria in the presence of the metal or in the presence of a bullet, and you would do these what we call serial passages. You would change the media daily for the bacteria, keep it fresh, keep it in this stressful environment in the presence of a heavy metal. And then what we did, we took that bacteria after a while, and then we started assessing its antimicrobial susceptibility. And if you guys would actually look at this table over here, you would see that resistance to levofloxacin was easily spotted whenever we grew an Acinetobacter baumannii in the presence of a bullet or in the presence of other metals as well. We've also observed resistance to Zirbaxa, which is one of these galactam beta-lactamase inhibitor complex that we were talking about earlier as well. We've observed as well plenty of other resistance mechanisms that I'm not highlighting here, but I would be actually happy to show you the conclusion, where what we have observed was when we created bacteria that were resistant to zinc and to cadmium, we observed resistance to cefepime. And the mechanism of resistance was an efflux pump component system that's activated by the heavy metals that targets the resistance to antibiotics. When we also made bacteria resistant to arsenic, we also observed the resistance to colistin, which is one of the last resort antibiotics. And when we sequenced, we actually figured out which gene actually encoded to the resistance of both. These are all novel findings that we are planning to actually finalize and publish soon. However, we didn't stop here, but we also started looking at clinical samples that could possess such properties, because we have to connect the dots. It's not, but we have to find it out in clinical samples. So what we have been doing, we've been looking at clinical samples that are resistant to several antibiotics, and then we've been testing those samples against heavy metals. And I mean, not surprising at all, we found that the more resistant to antibiotics the clinical sample is, the more resistant to heavy metals it is, and vice versa. So we know that this exists, but now we need to start connecting which heavy metal is becoming resistant to which, the bacterium resistant to certain heavy metals is becoming resistant to which antibiotic. And I mean, fortunately and unfortunately for us, the current conflict in Lebanon is giving us a big reservoir of samples. We've been collecting samples quite heavily lately, both on the clinical side and on the environmental side. And we've been targeting about 14 hospitals across the country and more than 6 bombing sites we've actually managed to sample. We've been looking at the bacteria in those sites, and we've been trying to understand a bit more this phenomenon in order to be able to pinpoint the molecular mechanism of how this can happen. With this, I will conclude my talk, and I would like to thank you for this opportunity again, and I'm happy.
Tony, I think you have to leave now, or shortly?
Yeah, I have to leave in 10 minutes.
So maybe any quick questions? I'm very selfish. I have one. So what do we know of the mechanisms of cross-resistance in these empirical data that you found? I guess, do you have any hypotheses? I mean, you mentioned efflux pumps. I think they're chelators. But in what you showed in your last or second last slide there, do you have any idea what's going on at the cellular molecular level here?
So we do actually have an idea, and we've actually managed to pinpoint the efflux pump that leads to cross resistance when it comes to zinc, cadmium, and cefepime. This relationship between these two metals and that antibiotic. A little bit of more real-time PCR is needed to actually understand it a bit more and to be able to finalize. But the interesting observation that I am finding very interesting, and I very much like that idea, is when you incubate the bacterium with the bullet, we tend to understand a bit more, not only when you incubate it with the heavy metal. We're tending to get a lot more information out. If you look at the table over here, let me go back to presentation mode and pointer. This is Acinetobacter incubated with a grinded bullet or with a whole bullet, it doesn't matter. But if you would see, there is resistance to fluoroquinolones, in addition to these combinations of beta-lactam, beta-lactamase inhibitors. Sequencing has not been done yet on those two in order to understand what really changed, but this will be done very soon. And we know for a fact that if you incubate it further, so this has been ongoing for, it went on for 3 months, then we stopped because of the financial crisis and COVID, but then we carried on with it for another 3 months, and we've observed even a bigger change in the antimicrobial susceptibility. Now we have something which is MDR, Acinetobacter baumannii. It went from fully susceptible to MDR just by incubating it with a bullet for 3 months. We hypothesize it would be cross-resistance more than any of the other two mechanisms. We are yet to prove it.
Bullets are, excuse my ignorance, but they're polymetallic.
Yes. Yeah, they are polymetallic.
Okay, so and coming back again to, I think I see this is fascinating and great work, obviously, but I think there's 3 or maybe 4 levels here. There's this laboratory work, which would allow or points towards uncovering molecular mechanisms, and this is why you need the sequencing data to sort of get the structure of the protein, right?
Yes, to understand at the molecular level what's happening.
At the clinical level, I think this is a little less clear because bullets can stay in people for 3 months, and then they can develop films. What they call them, biofilms, and this kind of thing. But there's a lot more going on in the clinical level because people are being treated with antibiotics and so on. But it's very compelling. The environmental. I'm just wondering to what extent are the clinical organisms present in environment? Oh, I guess they're just exchanging genes, basically, right? So Louis-Patrick, maybe we'll talk about this. So I retract my question. With respect to the molecular mechanism of cross resistance, is it because there's a common binding site, or I'm just curious.
Yeah, so it could be because there is a binding site. It could be because there is a common way the bacteria gets rid of what causes stress on it as well. Mm-hmm. It could be either or, and it could be both as well. But coming back to your previous question that you retracted, I would just like to point out between parentheses that quite a lot of the clinical samples that we find in patients we're also finding in the environment. When we did the surveillance of AMR in the country not so long ago, where Anthony was with us, and he actually helped us quite a lot. We looked at AMR in sewage, in soil, in wild animals. A lot of the carbapenem-resistant gram-negative bacteria that we found were actually samples that we don't only find in the environment, but we were also finding in patients.
Okay. Any other questions before we release? It's super exciting to see where you're going with this. Wow. And
it's very scary to see Zerbaxa on your list because it's not that old as an antibiotic. It's quite a relatively new one, right?
Yeah, well, it depends how you see it, as old or new. For me, Zerbaxa is as old as any of the other beta-lactams, because Zerbaxa is not very far from augmented, because the way it works, it's a beta-lactam with a beta-lactamase inhibitor, and we're still stuck in the same concept. This is what I was trying to point out.
Right. Yeah.
Yeah, this is what I was trying to point out earlier about. Yeah.
Any rationale? I know you've sort of discounted this idea, but any rationale to looking for phages in the environment here?
So there is another faculty member actually benefits from all of this. He has an interest in building phages of libraries of phages for clinical purposes. He's been doing this work, but we've been trying to, with his help, to look at phages that could actually cause the resistance instead of combating the resistance. And at the moment, up till now, we haven't been very successful, but I think we were doing it the wrong way. So now we're changing the approach.
Okay. I don't know if Aula or Louis-Patrick, if you had any questions. I'm not online on my computer, so I can't see if there's the chat box. There's no, okay. Okay. So, thank you very much.
Thank you.
And, well, I guess we say stay safe, right?
Yeah.
Thank you.
So.
It was lovely to see you all, and we'll be in touch. And I apologize, I have to go, but.
No, we understand. We appreciate your having been here with us so far.
Yeah. Thank you.
Thank you.
So our next speaker is Dr. Aula Abbara, who is going to talk about AMR conflict and humanitarian healthcare. Aula is a consultant in infectious diseases, honorary senior clinical lecturer at Imperial College, and has just been with MSF in Uganda and Yemen, was it?
Ethiopia.
Ethiopia. So over to you, Aula.
Thank you, and thank you to FINPHARM and Tony and the rest of the team for inviting me. I'm having a little trouble sharing my screen, but I'm hoping I'm getting there. Now, can you see my slides okay?
Yes.
Fab. It means
Perfect.
I can't see you, so feel free to shout out any questions as I go on. And I talk way too much. I'm going to try and keep to time to make the most of Louis-Patrick's presence as well. It's always wonderful to listen to Tony and the rest of our colleagues speak about this topic. And I'm sure you're aware of some background, which I won't say exactly when because it will age us, but some years ago, Kim, Omar Diwachi and others started to have dialogue about the sort of current evolution and impacts of conflicts at that time predominantly in we don't call it the MENA region anymore. I think we're trying to call it the WANA region at the moment, and how they interrelate to antimicrobial resistance, whether through Iraqibacter, Acinetobacter, or other resistant bacteria. And so what I was going to do today is try and capture perhaps some of the broader work that we've been discussing as a group, and it's actually a very similar group to the one that FINPHARM initiated, with some new faces bringing fresh ideas and discussion to this really important topic. And so what I was going to do is introduce what we're in current discussions trying to form into a network, a little bit more about AMR and conflict, building on the excellent work that Tony has just been speaking about, what innovations there are in terms of trying to tackle AMR in some of these incredibly difficult situations, Gaza, Syria, Yemen, Sudan, which are all quite different, I think, from the work that's being possible at the AUB in comparison. Though, of course, there are major challenges in Lebanon at the moment. And if there's time at the end, I'll talk about innovations and future work, if that was possible. And so on the back of the previous discussions, which started with the ERC grant a few years ago, where we brought together an interdisciplinary team of epidemiologists, clinicians, surgeons, social scientists, environmental specialists, microbiologists, diagnosticians and others, to bring their heads together and discuss some of the theories and pathways and development around antimicrobial resistance. We've put this figure, well, I've put this in grey at the moment, and I've shrunk it a little bit because it's in press, but I wanted to show to you some of the start of how this network of people, many of whom you know, so people like Omar Diwachi, Richard Sullivan, who've been mentioned, Nabil Karraf and others, have brought to this discussion. And when I talk about AMR and conflict, I do two things. One, try and emphasize that AMR and conflict is different from the conversation about AMR and low resource settings. There are different drivers, as we've heard from Tony. There's different challenges, as you'll go on to see. And there's also different impacts and probably a mix of similar but different ways to try and tackle this issue at multiple levels, on the personal level, the health system level, the community level, the environmental level, and then I can go onwards. So I try and bring across this element of interrelatedness and complexity, and because of some of the work that I do predominantly on Syria, but also some of the other humanitarian crises through my other work, my work with MSF, is thinking more about perhaps some of the regulatory or political determinants, bringing in some of the climate, disaster and environmental determinants, and that goes beyond just this discussion around heavy metals. It goes on in terms of what are the bacteria contaminating effluent from hospitals in our environment when we don't have enough water and sanitation. So what are we using clinically to clean wounds? And of course, this is a major issue, particularly in Gaza and Lebanon, but also elsewhere. And then this additional human factor around demographic instability, overcrowding, living in tents, and the impact all of that has on wounds, wounding, contamination, healthcare access, antimicrobial use, and then onwards. So we're in current discussions around forming this network, deciding what we're about and where we will sit. And it's very likely that this will sit at Imperial College. One, because there's some excellent antimicrobial resistance initiatives, particularly the Fleming Initiative, which has just been established very recently, which has a particular focus around community engagement, community impact, but also the sharing and discussion of work exactly like this. So as part of this network, one of the things that we've done is submit this paper, and you'll see the title there, and you'll see many familiar names who've put their minds to this. This is an early prototype of the figure, and I think we'll go on to develop it. And what I've taken away is some of the peripheral things, which bring in some of the other factors which are also important to discuss whenever we are talking around AMR and conflict. The next thing I do when I talk about AMR and conflict is also try not to dissociate it from the fact that many conflict-affected settings are also affected by natural disasters, by climate change, by forced displacement, and all of their impacts on population, healthcare access, and by extension, antibiotic use, antibiotic quality. And this was a really important thing that Tony highlighted, which is hugely problematic in Syria, but also how wounds develop, how antimicrobial resistance develops in these settings, and how that all relates with conflict and all the different sorts of conflicts that we see in their complexity, in their impacts, in their wide-scale destruction with civilians, but also armed combatants, having different risk factors for antimicrobial resistance in different ways that they access healthcare services. And so I increasingly discuss AMR and conflict, and I presume you can employ this paradigm to almost any health condition or infectious disease, which is what my specialty is, in all the different complexities. And so when we think about these complex emergencies, we're talking about political instability, environmental instability, economic instability, and this is important because it's not just about health system economics and financing. For us in Syria, this is hugely problematic because not only is the health system as a whole underfunded, our health workers and therefore their motivations may be underfunded, but also our ability to establish functioning microbiology laboratories with the right quality control for verifiable results, as Tony showed, are also affected. And of course, poverty, particularly intersectional vulnerability, so we know that women may be more affected by antimicrobial resistance, also come into play here. And often, and you might see with some of the WHO Euro work, that refugees and migrants may be affected or may have different behaviours that are relevant to antimicrobial resistance. And you'll see that at the UN high-level meeting from a few weeks ago, there was a big push, particularly around appropriate antibiotic access, because having too much and too little antibiotics in themselves can be problematic. And then lastly, of course, demographic instability and how that relates to conflict. And so in summary, when we think about AMR, we can't just think about conflict and AMR as a binary relationship, but also thinking about it in terms of natural disasters, displacement, climate change, but also particularly with a One Health perspective. So for example, there's massive antibiotic overuse in agriculture or in farming. And of course, the effluent of that can go into the water systems and therefore affect our populations. And I think it's important not to ignore this. And of course, there's a separate conversation around the impact of heavy metals and how they relate to antimicrobial resistance. I think many of you will be familiar with this, and I won't speak about it in too much detail, but I always come back to this. And I think very sadly for many of us, we are living history, and we are living the current pressures that could drive not only more antimicrobial resistance, but also catastrophic impacts to our humanitarian systems. And the takeaway from this is two things. First and foremost, for clinicians such as myself, it's been really important over the last few years, and that's particularly thanks to Kim, Omar, and many others who've been part of these networks, is to go beyond the biomedical or the biophysical approaches to how we address antimicrobial stewardship, infection control, and the different pillars that we deal with, particularly in health systems where we don't have the privileges that we have in some of the health systems that I work in. I work down in London, where we have high functioning laboratories, PCRs, and almost anything else that I can request. And so all of this ties into this important discussion around war's toxic legacies. And for me, that goes beyond the environmental, it goes into the geographic, into the human angle, into the health system angle, and then onwards. And sometimes, and you would have seen Tony's slide where in Syria, we don't really contribute anything in terms of appropriate surveillance and antimicrobial resistance data. Having said that, we have published some data, and I haven't shown it here, from areas under government control. There was a problem with quality, but with the data, and it was a few thousand samples from some of the hospitals in Damascus and Latakia and elsewhere, we did see very high rates of antimicrobial resistance, albeit with some questions around the antibiotics they were testing and the MIC interpretations for certain bacteria. I do sometimes despair, despite the work that I do on Syria, particularly the northwest and the northeast of the country, which are the areas outside of government control, where the health systems have been devastated and continue to be devastated by the conflict, with attacks on civilians even this week. And I just wanted to show you the reality of what the healthcare workers in these areas deal with. It's the chaos, it's the complete understandable inability to have any sort of infection control. It's the peak in the wave of an attack before you might have a period of relative calm where you start to run into problems with your external fixator infections. Sadly, at the start of the conflict, the behaviour of the surgeons who are not familiar with war surgery, they were putting in internal fixators into open wounds. And those of you who are familiar with this will know that's a surefire way to generate infection and osteomyelitis, which will be very hard to deal with. and also approaches to surgery whereby people may attempt limb salvage with the best of intention. However, there is no chance the bones are going to heal. And secondly, you're really just delaying the inevitable, albeit having to treat infection after infection for some of these patients and potentially driving antimicrobial resistance in the wake of this. Syria, and I won't speak too much about the conflict, but can do if there's more time. We've entered our 13th year of the conflict, and at the moment it's possibly at a frozen conflict, but there's been an escalation of attacks. And at the same time, we've also seen significant destruction of healthcare facilities. Very sadly, in Gaza, we see the same thing. So the majority of health facilities and all microbiology facilities have now been destroyed. And I'll speak more about that in the next slide. And that becomes really catastrophic in terms of knowing what you're dealing with, whether at the start for your empiric antimicrobial treatments, or what you're dealing with at day 2, day 7, and in the longer term for the sake of these patients. I wanted to highlight here some excellent work from Claudia Trapper. So myself and some colleagues have published some work, usually small case series, around antimicrobial resistance among Syrian refugees, cases from Syria, etc. But this is a lovely review that Claudia, who is the ICRC infectious diseases specialist, has put together. And actually her headlines are key take-homes when we look at Middle Eastern conflict. Well, she looked at conflict and non-conflict affected settings. And the key take-homes here are underlined. So CRE, so carbapenem resistant rates for Acinetobacter are very high. Carbapenem resistance for CRE, sorry, here it's E. coli and Klebsiella, are also very elevated, as are ESBLs, as is MRSA in these populations. We've just completed another review, and I'm afraid I didn't have time to put the data together, which we've submitted, which looks at conflict-associated wounds in a number of countries across the Middle Eastern region. So Iraq, Jordan, because of the impact of Syrian refugees, Lebanon, Gaza, and Syria itself. And again, what we see, one is sparse data, 2, poor quality data, and 3, what data there is shows that there's incredibly high rates of resistance, particularly to gram-negatives, less so to the gram-positives like Staphylococcus aureus. And the other piece I wanted to highlight here, and I'm sure you're familiar with it, but it's also a talking point here for Gaza. So as of February 2024, there are no functioning microbiology laboratories anywhere in Gaza. At the same time, we know that we are, I think the latest figure is about 100,000 people with limb injuries and amputees and catastrophic life-changing injuries. A devastating fact is also that we've probably got the largest child cohort of amputees probably anywhere in the world. What that means, other than the humanitarian and emotional and human impact, is these are children that are going to face, for some of them, problems with long-term infections, obviously the need for prosthetics, and as children grow, it's not as simple as if you're an adult that needs a prosthetic limb. So that's going to grow with them, and it's potentially going to put them at risk of long-term complications which relate to infection and the potential for AMR. And I say the potential for AMR, but actually Crystal, who's the MSF Lebanon epidemiologist who is doing a lot of work around this, put some of the MSF data between 2018 and 2022 before the current attacks on Gaza. And what she showed is incredibly concerning in terms of multi-drug resistant organisms, which are upwards of 70%, with the majority of the samples collected having osteomyelitis, so chronic infections, difficult to heal, bearing in mind that bone doesn't really have a very good blood supply. They've also done some other work, which I hope will come out soon, looking at Pseudomonas resistance and multi-drug resistant Pseudomonas. And those of you that may be aware, Pseudomonas is a relatively indolent gram-negative. It really does like metalwork, it likes biofilm formation. And it becomes really problematic when it gets into a wound and is causing infection. And you'll see here at the bottom that carbapenem resistance rates are 25%. So sometimes I get asked about patients in Syria and in Gaza who are not getting better on carbapenems, which is one of the last line antibiotics that we're able to use. And again, sometimes I feel that we can do more in Syria, that we should do more. Very sadly, I could probably show you these photos in Gaza. So this was M10 hospital in Aleppo. So in Syria, we've had well above 600 attacks on healthcare, and they are ongoing, and more than 900 healthcare workers have been killed. And we've done a lot of training over the last few years, but particularly in the last couple of years, to support our microbiologists and microbiology technicians. One thing to note just very briefly, when you have conflict, microbiology, diagnostics and things like this, perhaps because AMR is quite insidious compared to a gunshot wound or the need for amputation, etc. It's much more insidious in the way it affects people and the way it kills. It's often neglected, particularly where you have an underfunded humanitarian system and humanitarian response. So this is one of my colleagues that we worked with when he was in Aleppo, and below is you'll see that it's the theatre. And this was a hospital, I can't remember how many beds, but it had an ITU, it had I think 2 operating theatres. And then this was the next photo at the end of that year, where the whole hospital had been razed to the ground. And sadly, this attrition of building laboratories and finding that you can't access them or they're bombed is not an infrequent story. And again, I think it's also important for us whenever we talk about conflict, particularly about AMR, is to remember that many of the health worker colleagues who are very capable, and you'll know that in Gaza, one of the heads of public health and heads of the laboratory was also killed in the last few weeks. And this is Mohammed, who's one of the microbiologists that was killed in November of last year in the course of the conflict, along with several of his other family members.
Well, we just have a few more minutes for your presentation.
Cool. I'm going to try and speed up a little bit,
Yep.
and I'll just highlight a couple of things. So obviously there's the microbiology stuff, the epidemiology, and lots of the other things which are very important for us to understand. I've become increasingly interested in the human stories, and probably from the influence of Umar and Vin Kim around how patients, many of whom are vulnerable, interact with antibiotics, interact with the health system, what their experiences are like. So this is a piece of work with crew at the AUB, the American University of Beirut Center for Research and Education in the Ecology of War, where one of my supervisees, Ahmed, interviewed patients themselves affected by conflict-associated wounds who've been affected by AMR. And I'd urge you to read this paper because it's sort of a touching record of what they underwent, but also you'll begin to see the delays in their treatment, the challenges that they have in terms of but I've increasingly been asked to support around Gaza. And it's a number of different things. We've got more work around developing the baseline, supporting the laboratories, and we're making some good headway with that, aiming for good quality, supporting where we can in terms of the pillars around antimicrobial stewardship and infection control, and then supporting the sort of rebuilding, particularly in the northwest and the northeast of the country. And this is much more of the, less the academic arm to an extent, much more of the response arm. In terms of laboratory strengthening, I'll cover this super briefly. I think Tony highlighted the key issues. This is one of the laboratories. The top photos are from Raqqa and the bottom are from northwest Syria. And you see...
we're not seeing your screen.
How strange. Okay. Shall I try one more time?
I think you banked out, but we're almost out of time too, so okay. Maybe...
Shall I could probably... Do you want me to stop, Vinkim?
Can you wrap up?
Yes, I can do. So, for some reason, let me just see if it will let me share. Sorry, I'm trying very hard not to talk too much today. Fine. Let me just show you these photos because it'll give you a bit of an impression of what we're dealing with. This is one which is around telemicrobiology and humanitarian crises, which was led by Nabil Karrah to support some of the microbiology services in northwest Syria. And then this is the MSF mini lab microbiology services, which I think many of you will be familiar. And the idea is very quickly you can deploy this, set it up within a matter of days, and have it functioning even in the most challenging of circumstances. And this is the Antibiotic app, which is where if you have insufficiently trained microbiologists and microbiology technicians, you can use AI to support the reading of MICs and support therefore more appropriate antimicrobial use and stewardship. And I'm going to stop there, and these are the key messages here around the differences between conflict and disasters and low resource settings in terms of antimicrobial resistance, the need for multi-stakeholder approaches, the need for innovations, and I think lastly, the need to listen to our patients, communities and health workers if we really want to understand this issue around AMR in conflict settings. And I will stop there.
Thank you, Aula. You are able to speak quickly and clearly, so you get a lot of information across. That was great. We'll go now to the other side of the Atlantic. Louis-Patrick Haraoui, who's an infectious disease specialist, associate professor in the Department of Microbiology and Infectious Disease at the University of Sherbrooke. And he is also a member of the Canadian Foundation for Advanced Research, Human and Microbiome Program. Over to you, Louis-Patrick.
Maybe we can have some questions with Aula.
Yeah, Aula, any questions for Aula while Louis-Patrick... Yes.
The technologies you were starting to show towards the end, Aula, I was wondering if you could speak a bit more about their financing, their development, their deployment. Really extremely curious.
Absolutely. So I'll start with the MSF Mini Lab. And I'm not expert at this, but I have, so I don't do AMR with MSF, I do infectious diseases and malaria, but work very closely with my AMR colleagues. And it is, I'm not going to put a figure to it, but it's expensive compared to the basic microbiology that we may have in many of our settings. And actually, we're aiming for a basic set of microbiology machinery and culture for some of our laboratories in northwest Syria. And I can put a figure to that in the tens of thousands of dollars. The mini lab is more expensive, but I won't put a figure to that. And then in terms of the telemicrobiology, we were able to implement it with a grant from Canada Grand Challenges. And I don't think that was a large grant in terms of implementation. You saw the idea was it's a porta cabin, some simple microscopes, some simple camera phones, simple microbiology cultures and discs. So the costs of the consumables are not so great. What I didn't mention was that with the telemicrobiology project, we were able to make some good headway. However, we were challenged by the fact that with active armed conflict, we were no longer able to reach some of the areas where we set up the telemicrobiology facilities. And so when I do visit these areas, I'm often told how great an initiative that was and how they would like more of that. So that feels like an important technology to develop further. For the Antibiotic app, I don't know if, Louis, feel free to speak up whenever you want to test the microphone. The Antibiotic app is, I don't know the costs of it, but I do know that they've made it free in terms of the technology. So that is something that can be rolled out. But of course, you do need your microbiology services because it's all about reading MICs.
And what about consumables for the mini lab? How does that work?
So I don't know the cost of those, but I'd imagine they're in the same similar sorts of costs to any sort of laboratory. The mini lab costs are mainly related to the incubator, the safety hoods, the blood culture, machinery and things like that. So those initial costs.
Great.
Can you hear me?
Thanks. So I think, Louis-Patrick, we can hear you now.
Oh, fantastic. Sorry about the...
Over to you. You have 20 minutes, and then we'll have maximum 10 minutes of questions, then we'll have a 15-minute break for tea.
Great, thank you. Are you seeing the slides in presenter mode or?
Yes.
Or, okay.
Full screen mode.
Full screen mode, perfect. All right, so thank you for the invitation, for the opportunity to present. I have no conflicts of interest to declare. I just want to list out a few objectives for the talk. So I want to go over some of the causes of antimicrobial resistance. I won't dwell as much on this because it's been so well presented by both Tony and Aula, but kind of touch a bit on some of the links with armed conflicts and talk about how we can leverage artificial intelligence to detect new sources of AMR. So I would say in about the last 15 years, there's been quite a shift in our understanding of AMR, in that initially we were mainly focused on the clinical setting as a site of AMR emergence and spread. And although that's still very much an issue, we've come to realize that most of the sources of resistance, whether it's bacteria that are resistant or of genes that encode resistance, actually come from the environment. And actually that makes a lot of sense in many ways, because the majority of bacteria, of microbes, are actually found in the environment. If we look at the different distribution of microbes, we see that they're predominantly in various soil or oceanic environments. And these environments are constantly being exposed to pollutants. We talked about heavy metals, biocides, many other things. And so resistance develops in these environments because bacteria have to adapt to these polluted environments. So in fact, when we're talking about AMR, the clinic is actually just the tip of the iceberg. Most of what is happening in environmental settings. The other thing that I will briefly touch on is that, in fact, it's not just that bacteria can circulate between these different ecosystems. It's also that bacteria have this special power, this kind of bacterial sex that they have, in which they're able to exchange genetic material. So if a bacteria, as the one on the left, has an extra piece of DNA that encodes a resistance gene, it has the superpower. It can actually spread that superpower to its neighboring bacterium, which enhances the spread and the dissemination of resistance. We've talked already about this. I won't go over this in detail, but as Tony highlighted, heavy metals are another means through which bacteria are have enhanced their ability to become antimicrobial resistant. And we've talked about heavy metal pollution in armed conflicts, but I think one of the things that's important to keep in mind is that this pollution can linger on for decades. This is the first article I'm listing is a group in Belgium that sampled soils around Ypres, which was the site of a famous World War I battle, and found that even about a century later, there were very much higher concentrations of heavy metals. So it's not just that the heavy metals remain, it's also, in fact, the impact that these heavy metals have on the soil bacteria, for instance, that are there. And as Tony also mentioned, there have been increasingly this awareness about this, and these post-conflict environmental assessments that have been done, as this one was done in Lebanon in 2007, following on the war that took place there in 2006. So how do we link antimicrobial resistance and armed conflict? Well, one of the ways is through this pathway that was already discussed. And we can actually see that if we look in alternative settings, let's say the beautiful Scottish Highlands, we can look at historical soil samples, so soils that have been accumulated over time and kept in soil archives. And when we look at the abundance of resistance genes in these soils, we actually see that there's a correlation. So we have this theoretical correlation that we know about, that heavy metals promote antibiotic resistance, but we have this empirical data also to support this. And also this paper that Tony, Aula, Vin Kim, myself, and I worked on a few years ago and published looking at specifically Acinetobacter baumannii. So if we take a step back and look at the bigger picture, what we then can assume is that there are innumerable and as of yet unknown antibiotic resistance genes in nature, and that these resistance genes can spread to bacteria that then infect humans. And so what we want to try to do then, it's not, I mean, there's all this great work that Tony and Aula mentioned, which is really focusing on the consequences and the direct impacts. But the work I'm mostly focused on is trying to see if we can prevent the increase and the spread of resistance. So here what we see is, for instance, bacteria that are in different ecosystems or associated with different hosts. So some are in soil and water, some are in animals and sewage, and others in humans. And when there are interactions between these ecosystems, so an animal drinking water or a human spending some time with animals, we can see that there's these opportunities for these resistance genes to spread. And the question is, can we prevent these spread events? And so there's been quite a few people who've looked at this, and the way they've done this is to use an approach that one would call comparative genomics. So they say, well, if we know the gene sequence of resistance genes, so if we know what they, how they're encoded, we can probably find novel resistance genes because they will have a similar genetic sequence. This is the same kind of work that some people have been doing around phylogenies and genomics, trying to compare different species between each other or even people within a single species. And this is an example of what a resistance gene phylogeny might look like. So this is a resistance gene that I've spent quite a bit of time studying, NDM. It's one of the most problematic resistance genes on a global scale. And what we actually realize is that this idea of doing a comparative genomics is actually not that useful, because when we compare NDM to one of its closest relative, they only have 32% identity. So not that promising, and I'll try to explain why I think we can have a different approach that's more successful. So just for the non-biologists in the crowd, just kind of a recap, just to tell you that a gene encodes what's called a protein. So when we talk about antibiotic resistance genes, these genes lead to a protein that is active against an antibiotic and can either destroy it or modify it or something else. So how does that work? So a gene is encoded in DNA. It then gets transcribed into RNA. So a lot of you have heard about mRNA these last few years with mRNA vaccines. And then the mRNA encodes the protein. And the way it's initially encoded is what's called an amino acid primary structure. So it's just a chain of amino acids. But in fact, that primary structure of amino acids actually undergoes various conformational changes and then ends up assuming what's called a tertiary structure, or in some cases a quaternary structure if there are multiple proteins that are aggregating together. So these are things that are in 3D and actually are quite unique depending on what the primary structure is, and the primary structure is itself dependent on the genetic structure. So comes along this great tool called AlphaFold. Some of you may have heard about it. It was quite in the news in the last few weeks. So AlphaFold was a protein structure prediction tool that was developed by an artificial intelligence group within Google. And the reason this has really revolutionized biology is that trying to determine the tertiary structure of proteins, so that kind of 3D conformation I was mentioning, used to take up to 5 years to do for a single gene. It was painstaking, it was a lot of labor, and sometimes it could take a PhD student his whole PhD to decipher a single protein structure. Came along AlphaFold, and they have been able, so within a few minutes, you can now detect and predict a protein structure. So quite a revolution. And now this tool is widely used by researchers around the world. to the extent that the people who developed this were recently awarded the Nobel Prize in Chemistry. So if we go back to this idea of protein structure determination, I mentioned to you these two resistance genes earlier, NDM, which is the one that kind of the new kid on the block that appeared and that has become so problematic. And I mentioned to you earlier that its closest cousin in some ways was this resistance gene called VIM. Now, if we do the 3D structure of these two proteins, this is what they look like. And what we can do is overlap them and see that, in fact, they tend to look quite similar. And if you recall, I told you that they only looked alike to the extent of 32% of their genetic or amino acid identity. But when we assess their similarity through something called a template modeling score, which is a score where we try to determine the similarity of two protein structures, they actually are almost identical. So this template modeling score goes from 0 to 1. 0, you are completely different, and 1, you are essentially identical. And anything above 0.5 is considered significant. So we're realizing, in fact, through these tools like AlphaFold, that very different genetic sequences can lead to very similar proteins. So why is this important? Well, it's making us realize that these cutoffs that we have to try to determine and identify new resistance genes were completely overinflated, and that we needed to consider similarities that are much lower. And so we've decided to apply this approach to a lake in Kolkata, India. This is a lake that was sampled by another group a few years ago, and they determined the genetic content of bacteria found in that lake. And the reason we focused on that lake is because it had a high heavy metal content and was also surrounded by pharmaceutical industries, with the potential that some of the effluent from these industries was finding its way in the lake. So what did we do? We took these genetic sequences that this group had done. So these bacteria found it in the lake. We determined their amino acid sequence identity. Then we determined their protein structure, and we then compared the template modeling score that I mentioned earlier of these genes to existing resistance genes. And we took promising genes, so genes that we thought could be new resistance genes because they looked very similar to existing resistance gene. We put them in a bacterium, and then we tested them in the lab to see whether or not they actually caused resistance. So what did we find? These are just some of our results, so not all of our results. So we found at least three new resistance genes to a drug called trimethoprim, which is often used in combination with another drug of the sulfa family. And it's one of the most commonly used antibiotics in the world. It's first line for use in urinary tract infections, for instance. And what we found are these three proteins. Their tertiary structures are relatively similar to this existing resistance gene, but in fact, they only share about 40% amino acid identity. And when we line them up, they tend to have a template modeling score that's quite high. And we also found another resistance gene, this time to gentamicin. So gentamicin being another in another family of antibiotics. And again, very low amino acid identity, but very high template modeling score. So this is very exciting, very promising, but in fact, I'm only showing you the, let's say, the nice part of the results. We did end up testing a lot of other genes that did not turn out to be resistance genes. So there was a lot of false positives. So our approach, as it stood at the time that we did this initial testing, led to us thinking that there were more resistance genes in this lake than we thought. So we tried to see how we could improve our hit rate, how we could decrease our false positive rate. And so what we did was we decided to work antibiotic class at a time, so one antibiotic class at a time. And we decided to, this is an antibiotic class or antibiotic resistance genes, these AACs, they code for resistance to aminoglycosides like gentamicin, like I just talked about in our previous slide. And we said, what do all of the existing AAC resistance genes look like in terms of their sequence identity and template modeling score? And so we compared all of the existing ones. These are hundreds of existing resistance genes, and we compared them to each other. And we developed this XY plot where sequence identity is on the x-axis and the template modeling score is on the y-axis. And then we applied this thing called a support vector machine, which is a way of creating a boundary between the genes that look alike, or the resistance genes that look alike, and things that would fall outside. So this is our decision boundary. Everything that's contained within this boundary are these existing resistance genes. And then we said, well, when we identify new potential resistance genes, we will target them and see if they fall within this decision boundary, and to see whether or not that enhances our hit rate or decreases our false positive rate. And in fact, it has. We've gone on to identify some more resistance genes thanks to this, and so decreasing the amount of lab testing, which is really the bottleneck in our approach. It's much easier to do all of these computational steps, but when we go into the lab, there's all these technical aspects that are a bit harder to deal with. And then we've moved on to expanding the range of resistance genes or potential resistance genes that we'll be testing, including to beta-lactam antibiotics like the penicillin family of antibiotics, colistin, tetracyclines, etc. And this is the work of a really fantastic PhD student in my lab, Lachlan, who's been doing and leading all of this work. So how does this relate to armed conflicts? Well, I told you earlier that most of the bacteria that lead to these new resistance genes are found in the environment, that heavy metals contribute to increasing the chances that we will have resistance genes in the environment, because these heavy metals linger in the soil and force the bacteria in the soil, in the water, and elsewhere to adapt. And then we can say, well, if that is the case, can we actually detect these resistance genes before they make their way to human pathogens, before they start causing some of the problems that Tony and Aula told us about? And so this is a map of what's been going on in Gaza for the past year. We see that early on, there was obviously most of the damage was in the northern part of the strip. And as the conflict endured and continued, we saw more and more damage. And this damage is in fact not just the bullets, not just the shells, but all this infrastructure that's breaking down and contains metals and heavy metals in many cases, and is finding its way into different environments where bacteria thrive. And the question then is, are these destroyed environments becoming fertile ground for resistance genes, for new resistance genes to develop? And I think this is where there is a potential here to try to determine whether or not this is happening. And this is borrowing work from Doug Weir, who's a previous collaborator that most of us have been engaged with, and to leverage civilian science to see if we can use the ability of some of these people who are unfortunately living and suffering in these horrible conditions, and trying to see if we can get a way to access some of these degraded environments to detect whether resistance is appearing. So just to conclude, armed conflicts lead to increased risks of AMR. These are risks, and these new forms of resistance are concentrated in the environment, and these risks linger over decades. Combining genomics with AI tools such as structural biology enhances our ability to detect novel antibiotic resistance genes, and therefore to prevent these from spilling over into the clinic. And I believe that there is a potential for civilian science here to help us and to prevent the worsening AMR crisis that we are currently living in. So with that, I'd like to thank you for your attention, and I welcome questions and comments, and just some gratuitous advertising here that I am open to accepting positions as for graduate students and postdocs. Thank you very much.
Thank you, Louis-Patrick. That was great. I'm trying to just digest the presentation. So the last piece of work you did, the lake in India, the decision boundary. And so what you're saying is that what this work suggests is that relying on sequence alone underestimates the prevalence of resistance genes in the environment.
That's correct. So if we rely on genomics, which is the tool that people have kind of favored because of its ease of access, then we are underestimating the resistance, because we're missing all of these genes that look very different, but in fact, on the protein side are actually almost identical.
And just for the uninitiated amongst us, when we talk about, so tertiary structure is three-dimensional structure, and correct me if I'm wrong, that is determined by sort of chemical or structural bonds, but also electrical forces which fold things a certain way. And the quaternary structure is the folding and unfolding over time. Is that correct?
So that's correct. The only thing I would add is that the quaternary structure is mostly when you have multiple tertiary structures that are coming together to form a bigger protein. Mm-hmm. But there is, as you say, a very dynamic aspect to the conformation of these proteins, and chemical, electrical forces are at play, the environment in which these proteins are acting also. And that's why for such a long time, we were challenged in our ability to determine these 3D structures, because people didn't quite understand how we could determine how these simple linear structures would then conform in 3D. And now with AlphaFold and other similar tools, we're able to determine that in a much faster way.
I have some more questions, but I don't know if anyone in the online or here has some questions.
I do.
Yes, please.
Since you're going through the trouble to collect the samples, have you thought about looking for a collaborator to look for the drugs in the water themselves?
That's a good point. Not yet, but if you are interested or have people who might be interested, I'm more than welcome to discuss.
Yes, very good question as well. Okay, so the one question that I had is you're trying to break these contact areas, trying to identify the resistant genes in environment, or etc., before they get to the human. And how are you thinking that this can be stopped? Because that sounds the tricky bit, how to actually control this exchange.
Right. So that's a very good point. So there's a few things that I think could be done. One is that when we've identified these new genes, then we can start screening for them outside of these environmental settings. So we can try to see whether or not they can be present in bacteria that may in fact infect humans. The other thing is that if we know that there are certain drivers that are promoting the emergence of certain resistance genes, whether it's antibiotics that are spilled over or heavy metals or otherwise, then we can hopefully act on policies to argue for certain things to be changed about how these, let's say, promoters of resistance genes are acting. And I think the other thing is that we can actually link these with current efforts by pharmaceutical industry or others to deploy new antibiotics and say, look, there is an expanding pool of resistance genes out there. And if we want to deploy effective antibiotics, we need to take into account also these potential risks that are out there in the environment. So I think it's the slide I presented, obviously you're right, it does make it sound a lot easier than it is, but I think increased surveillance, altering policy, sharing with industry and other actors might be a combination of ways that we may impact and decrease the risks that they will spill over to human pathogens.
So it sounds like there's this real potential for anticipation and prediction in the downstream before the outbreaks appear clinically, let's say. Is that right?
Yes. So yeah, that's a very good point. So the challenge obviously is that the routine work that we do in clinical microbiology labs when we're testing bacteria, most of the time we're testing the ability of antibiotics to work against given isolated pathogens. But we're not looking on a molecular level what are the resistance genes that cause these. So for instance, the NDM gene I mentioned earlier, it was initially detected in 2009, but in fact it had been circulating for at least 5 years by the time it was detected, because people were just seeing a resistance profile but weren't assuming that there was a different mechanism underlying the existing resistance profile. So if we are able to anticipate new forms of resistance circulating and then spreading, we can actually have, I think, an upstream impact on the risks that they will spill over and cause issues in human settings and clinical settings.
I have just a quick question. The slide where you showed transfer between humans, environment, etc. There's an abbreviation HGT, which is that human gene transfer?
So horizontal gene transfer. Yeah.
And
that
is there not a marker of that? I seem to recall there's a trace. There's something that's found where a lot of horizontal gene transfer is happening. Can we look for that?
So
yeah. Is it transposon?
I'm guessing. We were thinking of, but transposons, are you thinking of?
Yeah, so this gene transfer is carried out by what we call mobile genetic elements. So these elements that can shift between these bacteria. And there are different forms of mobile genetic elements, and transposons are one of the most common ones that are carrying these resistance genes and spreading. So I'm not sure if that's what you were referring to in Kim.
Is there a marker of, I seem to remember Charles talking about this, that you can look for not the actual genetic elements, but indicators that there is intense gene transfer happening.
So there's recognition sites, right, on the ends of transposons you could probably identify, right, on the boundaries.
Yeah. So I think it's still a genetic marker, but it's mostly about so a lot of these mobile genetic elements have specific conserved sequences that enable them to initiate or a transfer between bacteria, or to determine where they will insert in certain genomes. So these are the traces that people look at, and these are much more conserved. So I went over it quite quickly in my presentation, but one of the things that we're actually doing, so in the lake samples, we're focusing on novel resistance genes that are already found on mobile genetic elements, because we think that these are at higher risk of spreading to human pathogens. So what we're realizing is if we actually look for all resistance genes, irregardless of whether they're found on mobile genetic elements, we're actually finding a lot more, which is quite scary when you think about it. But these at least have potentially a lesser risk of being spread because they're not yet found on these mobile genetic elements.
I asked the question, and that will be the last one because we have to break. Is there, we have this hypothesis about hotspots being in conflict being a good example of a place that favorizes hotspots for the emergence of AMR, which are environmental, all the things we've talked about. But that's kind of been a theoretical assumption, right? And I'm wondering if there's empirically a way to sample the environment, whether micro hospital laundry rooms or macro, to see where actually this is empirically happening.
Yes, and I think that's where the work that Charles and others have done around soils is quite interesting. So we have, I didn't present this, but we have a collaborator in Lebanon who provided us with soil samples from the late 90s and early 2000s, and who's offered to go back and resample the same sites. So we would have soil samples from the same area about two and a half decades apart. And we could look and maybe see if and these are across Lebanon, and we could maybe try to correlate. So we know, for instance, that southern Lebanon or the Bekaa or some of these other areas have been more intense areas of conflict and fighting. And if we can correlate changes in resistance gene content between different time points, then that may support that kind of empirical work to determine how these have been changed over time.
Okay, well, thanks. That was great. It was a great panel. We'll break now before turning to our closing panel with Ryan and Nadia about looping back to financialization.
Thank you so much.
Right on time.
We'll come back at 3:15 Geneva time.